Okay, let me see something. Oh, fantastic. Well, let us proceed then. Hi, I'm Dmitry, and I just want to see if this works or not, essentially. Uh, so I am a medical student, and I have to read a number of different textbooks, and I figured, hey, why not just stream it and see how it goes. So I'll proceed forward with this chapter, chapter 26, the urinary system, functional anatomy, and urine formation by the kidneys. Again, this is a PDF version of the Pocket Physiology book for Guyton and Hall. It is not the primary textbook. This is basically a a modified version of the textbook where it's a lot more condensed. So I'll be working from there today. Chapter 26. The multiple functions of the kidneys in the maintenance of homeostasis include the following: excretion of metabolic waste products and foreign chemicals, regulation of water and electrolyte balances, regulation of body fluids, osmolarity, and electrolyte concentrations, regulation of arterial pressure through excretion of varying amounts of sodium and water and secretion of substances such as renin that leads to the formation of vasoactive products such as angiotensin II. Regulation of acid-base balance through the excretion of acids and regulation of body fluid buffer stores. Regulation of erythrocyte production through secretion of erythropoietin, which stimulates red blood cell production. Regulation of 1,25-dihydroxyvitamin D3 production. Synthesis of glucose from amino acids, gluconeogenesis, during prolonged fasting. Secretion, metabolism, and excretion of hormones. And again, I just want to confirm that this is in fact streaming. Fantastic. So let me proceed forward. Physiological anatomy of the kidneys. General organization of the kidneys and urinary tract. The two kidneys lie outside the peritoneal cavity. Each kidney of an adult human weighs about 150 grams. The kidney is surrounded by a tough, fibrous capsule that protects its delicate inner structures. The two major regions of the kidney are the outer cortex and the inner medulla regions. The medulla is divided into 8 to 10 cone-shaped masses of tissues called the renal pyramids. The base of each pyramid originates at the border between the cortex and the medulla and terminates in the papilla, which projects into the space of the renal pelvis, a funnel-shaped continuation of the upper end of the ureter. The outer border of the pelvis is divided into open-ended pouches called major calyces that extend downward and divide into minor calyces, which collect urine from the tubules of each papilla. The walls of the calyces, pelvis, and ureter contain contractile elements that propel the urine toward the bladder, where urine is stored until it's emptied by micturition. So this is just a picture of the kidney, the ureter, the bladder, and the urethra, of course. Nephron, which is enlarged, minor calyx, major calyx, papilla, renal cortex, renal pelvis, of course, renal medulla, renal pyramid, capsule of the kidney. You get the idea, right? Section of the human kidney showing the major vessels that supply the blood flow to the kidney. And this is just showing the blood vessels right here, the arcuate arteries and veins, the interlobar arteries and veins, the renal artery, the renal vein, of course, the segmental arteries, the interlobular arteries and veins. And you can see here, this is a little more detailed, you could say. So this is showing the actual individual areas of the Bowman's capsule, the glomerulus, the proximal tubule, showing both the arteries involved as well as the actual part of the system that's doing things like collecting certain kinds of ions, collecting water, that kind of thing. And so you can see here, you can see the JGA, that kind of area where that releases renin. Depending on the situation, you can see the afferent and efferent arterioles. And of course, if you have a high GFR, it could be caused by something like, for example, You have vasodilation of the afferent arteriole as an A, afferent arteriole. You have a constriction of the efferent arteriole, the E arteriole. That can cause something like an elevated GFR, and vice versa. If you had a constricted afferent arteriole and a dilated efferent or efferent arteriole, it would have the opposite effect. So, but you can see, obviously, you can see the descending and ascending parts of the loop of Henle. You can see the proximal convoluted tubule. You can see, of course, the collecting duct where it ends. And of course, this upper part here is going to be in the cortex, and the lower part here, I believe both of the collecting duct itself as well as the lower part of the loop of Henle is going to be in the medullary region. And it just shows here the micturition of each nephron. Okay, back to the text. Renal blood flow constitutes about 22% of the cardiac output. Blood flow to the two kidneys is normally about 22% of the cardiac output, or 1,100 milliliters per minute. Blood flows to each kidney through a renal artery, which branches progressively to form the interlobar arteries, arcuate arteries, interlobular arteries, and afferent arterioles, which lead to the glomerular capillaries, where filtration of fluids and solutes begins. The capillaries of each glomerulus coalesce to form an efferent arteriole, which leads to a second capillary network, the peritubular capillaries, which surround the tubules. The peritubular capillaries empty into the vessels of the venous system, which run parallel to the arteriolar vessels and progressively form the interlobular vein, arcuate vein, interlobar vein, and renal vein. The renal vein leaves the kidney along the renal artery and ureter. The vasa recta are specialized peritubular capillaries that dip into the renal medulla and run parallel to the loops of Henle. The outer portion of the kidney, the renal cortex, receives most of the blood flow of the kidney. Only 1 to 2% of the total renal blood flow passes through the vasa recta, which supplies the renal medulla. Two distinguishing features of the renal circulation are 1, the high rate of blood flow, and 2, the presence of two capillary beds, the glomerular and the peritubular capillaries, which are arranged in series and separated by efferent arterioles. The glomerular capillaries filter large amounts of fluid and solutes, most of which are reabsorbed from the renal tubules into the peritubular capillaries. Okay, you want to confirm with that. So there's glomerular and peritubular are the ones that are filtering large amounts of fluid and solutes, but there's a lot of reabsorption in the peritubular capillaries. Got it. The nephron is the structural and functional unit of the kidney. Each kidney is about 800,000 and to 1 million nephrons, each of which is capable of forming urine. A nephron is composed of 1, a tuft of glomerular capillaries called the glomerulus, in which large amounts of fluid are filtered from the blood, and 2, a capsule along the glomerulus called Bowman's capsule, and 3, a long tubule in which the filtered fluid is converted to urine on its way to the renal pelvis, which receives urine from all of the nephrons. The renal tubule is subdivided into the following major sections, each of which has different structural and functional characteristics. 1, the proximal tubule, which lies in the outer portion of the cortex of the kidney. 2, the loop of Henle, which includes descending and ascending limbs that dip into the inner part of the kidney medulla. 3, the distal tubule, which lies in the renal cortex. And 4, the connecting tubule, the cortical collecting tubule, and the cortical collecting duct, which begin the cortex and run downward into the medulla to become the medullary collecting duct, which is number 5. Urine passes from the renal pelvis to the bladder, where it is stored until it eventually is expelled from the body through the process of micturition, that is to say, urination. Micturition. Micturition, the process by which the urinary bladder empties when it becomes filled, involves 2 main steps. Number 1, progressive filling of the bladder until the tension in its walls rises above a threshold level, which elicits in the second step. And 2, activation of a nervous reflex called the micturition reflex, which empties the bladder, or, if this fails, at least causes a conscious desire to urinate. The physiological anatomy and nervous connections of the bladder. The ureters carry the urine from the renal pelvis to the bladder, where they pass obliquely through the bladder wall before emptying into the bladder chamber. No major changes occur in the composition of the urine as it flows through the ureters into the bladder. Peristaltic contractions of the ureter, which are enhanced by parasympathetic stimulation, force the urine from the renal pelvis toward the bladder. The urinary bladder is a smooth muscle chamber composed of two main parts. Number one, the body, which is the major portion of the bladder in which urine collects, and two, the neck, which is a funnel-shaped extension of the body that connects with the urethra. The smooth muscle of the bladder is called the detrusor muscle. When the fibers contract, they can increase the pressure of the bladder to 40 to 60 mmHg, and therefore play a major role in emptying the bladder. The bladder neck, posterior urethra, is composed of detrusor muscle interlaced with a large amount of elastic tissue. The muscle in this area is called the internal sphincter. Its natural tone keeps the bladder from emptying until the pressure in the main part of the bladder rises above a critical threshold. Beyond the posterior urethra, the urethra passes through the urogenital diaphragm, which contains a layer of muscle called the external sphincter of the bladder. This muscle is a voluntary skeletal muscle and can be used consciously to prevent urination, even when the involuntary controls are attempting to empty the bladder. Okay. Pelvic nerves provide the principal nervous supply of the bladder. Coursing through the pelvic nerves, which connect with the spinal cord through the sacral plexus, are both sensory nerve fibers and motor nerve fibers. The sensory nerve fibers detect the stretch of the bladder wall and initiate reflexes that cause bladder emptying. The motor nerves transmitted to the pelvic nerves are parasympathetic fibers. The micturition reflex is a spinal cord reflex. The micturition reflex is a single complete cycle of 1. Progressive and rapid increase in bladder pressure, 2. Sustained increase in bladder pressure, and 3. Return of the pressure to the basal tone of the bladder as follows. Sensory signals from the bladder wall stretched receptors are conducted to sacral segments of the spinal cord through the pelvic nerves and then reflexively back to the bladder through the parasympathetic nerves by the way of the pelvic nerves. Once the micturition reflex is sufficiently powerful, it causes another reflex that passes through the pudendal nerves to the external sphincter to inhibit it. If this inhibition is more potent than the voluntary constrictor signals to the external sphincter, urination occurs. The micturition reflex is an autonomic spinal cord reflex, but it can be inhibited or facilitated by centers in the brainstem, mainly the pons, and several centers in the cerebral cortex that are mainly excitatory, but can become inhibitory. So basically what that's saying is, or let's say they give you an analog, you can have like a reflex in your muscles. Let's say, for example, I tap on your knee, on the patella of the knee, and I invoke for you to make a kicking, sort of motion. And that's a consequence of a reflex that's going to your spinal cord and then having a reflex where you kick out. It doesn't necessarily need to have the brain involved in that process. Now, of course, you can use your mind to kick like you typically would, but it can also independently have a sort of reflex without having a brain involvement. In this case, it's very similar. You can have a micturition reflex that is involved with the spinal cord without necessarily getting the brain involved, but of course the brain can be involved in micturition. Urine formation results from glomerular filtration, tubular reabsorption, and tubular secretion. The first step in urine formation is filtration of fluid from the glomerular capillaries into the renal tubules. As the glomerular filtrate flows through the tubules, the volume of filtrate is reduced, and its composition is altered by tubular reabsorption, the return of water and solutes from the tubules back into the blood, and by tubular secretion. The net movement of water and solutes into the tubules, each of which is highly variable depending on the body's needs. Thus, excretion of each substance in the urine involves a specific combination of filtration, reabsorption, and secretion is expressed by the following relation. Urinary excretion rate equals the filtration rate minus the reabsorption rate plus secretion rate. Did I get that right? Okay, urinary excretion is equivalent to filtration minus reabsorption plus secretion. So what they're saying is the amount that you're removing, that you're excreting, is equivalent to how much you're filtering out plus how much you're secreting, but removing all the stuff that you actually reabsorb back into the body. In general, tubular reabsorption is quantitatively more important than tubular secretion in the formation of urine. Let me repeat that. Okay. In general, tubular reabsorption is quantitatively more important than tubular secretion in the formation of urine. But secretion is important in determining the excretion rates. This is my fridge, okay? My freezer, I suppose. Okay, so anyway. In general, tubular reabsorption is quantitatively more important than tubular secretion in the formation of urine. But secretion is important in determining the excretion rates of potassium and hydrogen ions and a few other substances. Metabolic waste products that must be cleared from the blood, such as urea, creatinine, uric acid, and urates, are poorly reabsorbed and are therefore excreted in large amounts in the urine. Certain foreign substances and drugs are also poorly reabsorbed, but in addition, are secreted from the blood into the tubules, so their excretion rates are high. Basically, it's both. It's either being excreted, they're not necessarily being reabsorbed because it's poorly reabsorbing, and it's also being secreted simultaneously. Hence why you're removing so much of it in the urine. Conversely, electrolytes such as sodium, chloride, and bicarbonate ions are highly reabsorbed, so only small amounts appear in the urine. Certain nutritional substances such as amino acids, glucose, are completely reabsorbed from tubules and do not appear in the urine, even though large amounts are filtered by the glomerular capillaries. Yeah, the body is very, very keen on trying to preserve those and keep those in the body. And if you have glucose in the urine, that's an indication of diabetes, that's an indication of a pathology that's going on. It's not typical. Each of these processes is physiologically controlled, and changes in the excretion rate can obviously occur via changes in glomerular filtration, tubular reabsorption, or tubular secretion, as discussed in the next two chapters. Okay, and that completes chapter 26 of Guyton Hall's Pocket Physiology textbook. Thank you for watching.